Hello, my name is Emily Heaton, and I'm a professor in the Crop Sciences Department at the University of Illinois. It is my sincere pleasure to be with you today to share some of the work that's been happening at the Center for Advanced Bioenergy and Bioproducts Innovation, or CABBI, as we make progress towards using grasses as efficient, sustainable factories for bioenergy and bioproducts. This is a picture of the people of CABBI and where we are geographically located. CABBI is the newest of the bioenergy research centers and therefore could build on the excellent work that had come before us from our sister BRCs. CABBI is different from those other BRCs in that it focuses on the plants as factory concepts, with the idea that that would allow us to leapfrog some of the challenges associated with deconstruction and separation and go straight to converting compounds produced in the plants into desired fuels and products. Here's what this might look like in a pipeline kind of thinking. So on the left, we have Deconstruction Technologies, which I just said CABBI doesn't really do. And those come from the work that our sister BRCs have done. So we take what information is already available and we leverage that into our feedstock production, understanding growing bigger plants with fewer resources, like all the BRCs tried to do, but especially in CABBI using this plant as factories paradigm, where specifically we've been focusing on expressing lipids in plants to produce ready-to-use oils that can go into fuels, as well as into components for high-value chemicals. CABBI also is unique in that it relies on a biofoundry approach for our conversion platforms, where we create organisms that create new chemicals, particularly high-value chemicals. So the conversion and the feedstock teams work very closely together using genomic science approaches. You'll see work from both teams present today in the poster session. We use the same science to engineer plants and to engineer microbes that make the plants and the microbes produce the fuels and chemicals we need. And of course, underpinning all of this work is the sustainability efforts. And specifically in CABBI, we focus on analyzing the commercial feasibility of the products we described and their ecological impact. And another way to think about that in traditional Department of Energy terms would be the techno-economic analyses and the life cycle assessments. So let's look at that in a slightly more concrete way. In CABBI, we focus on some of the crops that you've heard about earlier in this session, particularly sorghum, but we also focus on sugarcane and miscanthus. And we improve those through the genetic engineering and breeding approaches that are common throughout this meeting into, as I said, drop-in fuels and byproducts. Then we have our conversion platforms that rely on the traditional approach of converting sugars and structural or free sugars into high-value products. And then we also have a body of work that focuses on producing lipids directly in plants and converting those into fuels and high-value products. So how do we decide which fuels and high-value products to focus? One of the things that I find really exciting in CABBI is this idea of integrated and agile techno-economic assessment with life cycle assessment built in. And this is the work led out of Jeremy Guest's group that really underpins our feedstocks, conversion, and sustainability themes. You can find BioSteam, the platform in question, at the CABBI website, then also documentation and directions for how to download and use this publicly available open source product at these websites. And BioSteam has now been incorporated into, I believe, nearly all the national labs and the bioenergy research centers. So this is really exciting because it lets us take questions about the type of feedstock we're going to produce and use, its characteristics, like for example, its lignin content, or ask questions about the type of platform molecule that we want to make or the type of microbe that we're using, or questions about the price of production of the crop or where we are in the United States or greenhouse gas emissions. It lets us ask questions about all of those things in a dynamic way with uncertainty. And so here's some of the example outputs that you can get from BioSteam, which again is publicly available and easy to use. So in this figure in the top left here, we have a a metric on the y-axis that's a techno-economic metric, in this case minimum ethanol selling price. And on the x-axis we have what's more traditionally a life cycle assessment metric, in this case global warming potential. And then the results that we get are focused in a target area like you might have seen normally. So let's just say we're looking at, in this figure, the production of biomass sorghum, for example, maybe biomass sorghum with altered lignin. And we're trying to understand whether or not there's a market for that biomass sorghum with altered lignin. And we're trying to understand how likely it is to reduce global warming potentials. So this figure would show us with uncertainty how likely our target crop and target engineering platform has made the improvements that we want to see. And so you can see summarizing bar charts of uncertainty here at the top. And similarly, we have metrics that relate to how our product would affect other markets. In this case, what if we were thinking about electricity markets? What if we were thinking about how the introduction of this, hypothetical biomass sorghum with lower lignin would affect things in both the US and in Europe? So these figures would show how energy might have changed depending on whether or not we were using the EU's metrics around life cycle assessment or the United States, depending on the different policies that are in place, and whether or not you'd have displacement in the allocation of energy based on the introduction of this, again, hypothetical biomass sorghum with altered lignin. We could do this for nearly any product that we talk about in CABBI. You'll hear us talk about triacylglycerols or some other target compounds that we're looking at producing in the plants as factory paradigm. And BioSteam lets us ask questions about whether or not we should produce those, how much money people are likely to make, how it's likely to affect the planet. It lets us ask those questions up front. Okay, so we have platforms like BioSteam, this agile integrated TEA and LCA, to guide our work in the feedstocks production theme within CABBI. But let's talk about the questions that we're really addressing in CABBI. So the challenge that we all face is that dedicated biomass crops typically only have one major end use, and that's bioenergy. And that leads to a decreased market flexibility. If you only have one place to sell your product, you're less likely to make that product. And therefore, plant biomass has had a low economic value, which increases the risk to any farmers who want to try something new. And then we've had inefficient conversion of lignocellulose to biofuels or bioproducts. It's tricky. So CABBI is founded on the premise that we were going to address these things by, first of all, trying to shift stem carbon from lignocellulose into more versatile and easily converted carbon forms. So think sugars and lipids. And that should help increase the value of the biomass from grasses that are already very high yielding. And we can also target that stem carbon into a diversity of compounds that are useful. We would also, like all the BRCs, have a focus on increasing yield efficiency and resilience in a changing climate. And specifically in CABBI, we have a focus on building high levels of oils and specialty chemicals and vegetative tissues following the plants as factory paradigm. So what does CABBI grow compared to the other BRCs? We grow grasses only, and in the eastern half of the United States. The grasses on which we focus are sugarcane, sorghum, and miscanthus. So you've already heard about sorghum, but sugarcane and miscanthus are very closely related grasses. All three of these genera have synteny between them, so they have their pros and cons in terms of their genetics and their ability to be transformed and their target growth geographies and industries. But really, CABBI produces grasses that we expect to grow on a range of lands in the rain-fed part of the United States. CABBI uses the design, build, test, learn loop that you've already heard about. Specifically, as I mentioned, when in designing, we're looking at using systems biology approaches to modulate gene networks, specifically focused on metabolism, shifting that stem carbon into useful forms of carbon. And we also focus on things even at the canopy and field scale, so working on a design approach that goes from subcell to whole field. We build new genetic variation as well as harness existing genetic variation. And then we test things, test phenotypes at, again, a subcellular scale all the way to the whole field scale. And then, of course, we learn from these things in our target crops. Specifically, the things that we are learning and testing relate again to lipids, to carbon partitioning, and yield efficiency and resilience. There are some key operational principles that guide strategic research in feedstocks. And to build on the CABBI analogy here, we use in silico predictions to map our way forward. So just like you would use Google if you were driving a cab, you got to know where you're going. We use in silico predictions and assessments to try to figure out the likely best paths to take when we are designing changes in our CABBI crops. Similarly, when you're driving down the road, it's good to look up and check traffic, check road conditions every once in a while. And so within CABBI, we do these deployments, demonstrations to test whether or not we think that the changes we're making in our crops are likely to work in the real world. So just to show you what I mean here, here's some highlights from a few recent papers showing the way we use in silico approaches as a framework to think about the likely effect that the changes we might make at a gene level, a cellular level, an organ or plant or ecosystem level might integrate together into outcomes. So we think about this a priori. We think about it in terms of metabolic networks, trying to use in silico assessments to understand what's likely to happen when we shift carbon from leaves to stems or shift it into different forms within leaves and stems. And then we do some field scale deployment to understand how the product that we plan to create or have created compares to what we have today and give us insight on likely hiccups that are expected to be experienced by a bioenergy industry. Like all the BRCs, we're happy to have a big and a vigorous team. We've organized in a few different ways, and I just want to show you with these slides that we think about organizing from discovery to end use. So working from, as I mentioned, this natural and engineered genetic variation all the way up, as you've seen with the other BRCs, through ecology and sustainability. And we do that with simulation, as I mentioned, but also association and manipulation of the pieces that we have with which to work in the feedstocks puzzle. We are also organized operationally because these are big projects and we have to break it down disciplinarily as well as from a product perspective. And so we have these overlapping teams with sub-team leads that really help us to make more rapid progress than might otherwise be expected. Okay, so what I'd like to do with the rest of this time then is to give you some examples, some illustrations of where we are, where we need to be, and how we are going to get there, as Kent said in his introduction. So to do that, I'm going to give you some examples that focus on meeting these challenges that I described to you earlier. I'm going to tell you a little bit about development of genomic resources that underpin doing all of these things in the CABBI crop Miscanthus. We'll talk about building high-value lipids in the CABBI crop sugarcane, so which we'll now start calling oil cane. And then, as I mentioned, we do these demonstrations. So I'm going to tell you about the oil cane pipeline demonstration we did last year. Then I'm going to tell you a little bit about stem preferred promoter candidates in sorghum. But you, I think, already heard some of that from Federica as well, so I'll just touch on that, but it's really exciting. And then I'll finish with describing some of the progress we've made in increasing the yield efficiency and resilience of CABBI crops, specifically highlighting some work in Miscanthus genetics and flowering time, and working on high temperature photosynthesis and ozone tolerance. All right, so starting off with those genomic resources for Miscanthus. Miscanthus is a new crop, and it doesn't have good genomic resources available for it yet. So last year, Matros et al. Published this paper in Nature Communications, and Teresa has a poster that I hope you'll visit during this meeting. But recently, we've built on the work that happened in that Nature Communications paper to really understand now not just about the genome, but where we have where we can expect to find genes on different chromosomes. So now we have a mix of both short and long read sequences that increase the confidence in the physical location of reads on the chromosome. And so this is just some lines of evidence that show that we believe the way that the chromosome is mapped. But what's really needed next to enable some improvements in Miscanthus is to be able to annotate this genome so we can understand the function of these mapped genes and target them for trait improvement. And again, I encourage you to see Teresa Mitro's poster. I mentioned that CABBI uses that design, build, test, learn loop. And the example I'm going to show you now is about improving trialysyl glycerol content, TAG, in sugarcane after 2 rounds of that DBTL cycle. So this is an example of why the design, build, test, learn cycle is increasingly common in some of our work because it leads to results. So I'm going to show you the example of oil cane that we recently showed in this 2020 paper that could get 8% trialysyl glycerol content in sugarcane leaves and 4% in the stems. So there's amazing progress, but so much left to do. So we'll continue to focus on accumulation in stems, and then we have to overcome some yield drag. And I'm going to tell you a little bit more about that, and then I'm going to talk to you specifically about some techniques that we're targeting to overcome that yield drag. And of course, we also want to optimize the specialized fatty acid accumulation that's happening in sugarcane, oil cane, and then translate that progress to other CABBI crops, next sorghum, and then as we develop the genomic resources for it, to Miscanthus. Okay, so the design, build, test, learn loop. For oil cane, that started off using Arabidopsis and benth, and has now moved to advance into to oil cane. So let me just tell you a little bit about the pathway that's being manipulated here, using this kind of nice handy alliteration of push, pull, protect. So the idea is that we want to push carbon into the chloroplast, into fatty acids. And so there's some genes that have been targeted to increase the amount of fixed carbon in the chloroplast that goes into fatty acid synthesis. And then from there, we need to make sure that there's not a buildup of those fatty acids in the chloroplast that would lead to a downregulation in photosynthesis. So we have to pull those fatty acids into new products. And now I'm showing you how it gets pulled into the endoplasmic reticulum in this case, where trialysyl glycerol is generated. And so again, a few genes of interest to focus on pulling those fatty acids through into trialysyl glycerol production. Once trialysyl glycerol is made, though, it has to be protected so that it's not broken down by the plant or again, lead to product accumulation that would downregulate photosynthesis or downregulate TAG production. So how to do that? Well, we protect the trialysyl glycerols in these lipid bodies. So push, pull, protect, and by the end, hopefully you have protected TAG that can be squeezed out of a plant and put into useful products. Okay, so that was the theory. There was in silico predictions and bench scale experiments in Arabidopsis and Benthiata that showed that this works, and it was put into sugarcane to make oil cane. And this was demonstrated for the first time in a time of COVID. As you can see from these pictures, this was quite the undertaking in 2020. But at 3 field sites, 2 transgenic and 1 wild type of sugarcane were tested. The 2 transgenic lines overproduced lipids. And then in a full test of the pipeline, the plant stems were squeezed, and the lipid content, the juice, was analyzed for lipid content at multiple labs. And then big gallons of it were taken to the Integrated Bioprocessing Research Lab and used as a substrate to produce high-value chemicals in the conversion group. So we're really testing all aspects of the CABBI pipeline here. And it was really exciting. We got really great results, but of course there's more to do, as is shown in this figure. So here's those 2 overexpressing lipid lines that I mentioned compared to the wild type sugarcane. And as you can notice just from the picture, as well as from this figure showing the biomass yield among those 3 types, there is yield drag, or a reduction in yield associated with the transgenic trait. So what to do? Well, first of all, we can find ways to increase trilacylglyceride content that also offset yield drag. And of course, as I think Heinrich mentioned, there's considerably more work to do regarding building the constructs for those, as well as targeting specialty fatty acid synthesis in those vegetative tissues. All right, so what can we do to increase trilacylglycerol production? Well, one idea is to increase the energy supply that it takes to make the fatty acids that make trilacylglycerol. As you all know, lipids are very energy-dense materials, and it takes a lot of energy to make them. So what about upregulating ATP supply? There's two genes of interest here that CABBI has been focusing on, purple acid phosphatase 2 and nucleoside triphosphate transporters. And I just illustrate here that one is associated with both the mitochondria and the chloroplast. The other one is associated with transfer of material between those supply chains. And again, there's a nice poster on this that I encourage you to see, the CAI poster. So does increasing the availability of ATP in oil-expressing lines of plants increase the trilacylglycerol that's accumulated? In short, yes. And the figure on the left here, where you have the purple gene expressed in higher amounts, there's an increase in trilacylglycerol when more ATP is available. And similarly, with the other gene tested, there is again an increase in trilacylglycerol in oil-producing lines when the ATP availability is increased. So increasing the push through the lipid pathway. Okay, shifting gears, I want to just highlight the work that Federica mentioned earlier, and that it's good to make lipids in plants, but it would be even better to make lipids in plants at the right time and in the right place. And so far, CABBI has had more success producing lipids in leaves than in stems. And so our approach to move that carbon into stems is to find stem-preferred promoters that can be used to trigger lipid production at the right time and the right place. And so here's an example of, I think, really nice collaboration between DOE labs and bioenergy research centers, building on the transcriptomic gene atlas of different sorghum tissues and different developmental stages that had been done previously, and then now moving forward to look within specific cells using laser capture microdissection to understand whether or not the genes that we think are in those cells are actually there, and to see if they're actually doing what we think they're doing. And as that work turns out, then we can move forward into expressing genes of interest in sorghum stem tissues. This work in CABBI is done in close collaboration with our in silico assessments to make sure that we're targeting the right metabolic pathways within sorghum once we understand if the genes work. All right, one of the other highlights I mentioned is development of genomic resources within Miscanthus, which is really exciting. But we also need to understand how Miscanthus flowers if we're going to backcross or in some other form get new traits into existing Miscanthus populations. And so this work is one of the first really big geographic ranging experiments that's part of CABBI. Similar to what you saw from the other BRCs. This is the work from Eric Sachs' group, along with a lot of international collaborations, testing the flowering time of Miscanthus types across Asia, also under controlled conditions, and really told us about what it takes to make different types of Miscanthus and saccharum species flower. And then lastly, I want to mention the work, which is again a large body of work within CABBI, focused on increased efficiency and resilience of photosynthesis. What I'm showing you here in this image is a sorghum growing in a ring of elevated ozone concentration in the field. And so this is work by many people, but specifically Lisa Ainsworth. And you can see that the corn right next to these pipes that's releasing the ozone is really unhappy. And this is why we study ozone. It makes plants really unhappy and causes a lot of yield drag that we might not otherwise notice. But hopefully you can see that there's a range of responses within the sorghum that's in that field. And so I encourage you to see the Lee poster today and multiple Lee publications on this topic. It's really exciting work that's been demonstrated in collaborations with other BRCs in model species, again, using that design, build, test, learn loop, as well as in now the CABBI crop sorghum. Similarly, there's been a large and exciting body of work in CABBI crops on increasing their tolerance to high temperatures. I'm not going to cover that in detail today, but I do have some citations for some of that work below, and I believe there's posters on that as well. So I'd like to wrap up by just highlighting that this CABBI is very fortunate to have the legacy of the other BRC centers on which to build, and we do. So I've been very pleased with the collaborative nature of these teams. These are fun places to work, fun activities in which to be involved, and really feel like we're working together to make a difference. And lastly, I just want to wrap up with some an illustration of how all of these things can come together around a feedstock pipeline. So this is a picture of a sorghum field that happened again in 2020, so a big body of work considering the pandemic, where members of the feedstock teams did various things within this field to alter carbon allocation in sorghum. In some cases, it was transgenic. In some cases, it was physical manipulation, such as bagging the flowering heads. The members of the sustainability team came out and sampled a wide range of soil and microbial biome traits that could be associated with this field. And then the juice from the sorghum was taken and is being used as a substrate in a wide variety of conversion experiments. So we've done, I think, a good job of integrating our physical and our scientific resources together to get shared understanding. And with that, I'll conclude, and I look forward to your questions. Thank you.